Hello students and welcome to the lecture of microbiology paper 1, that is Introduction to Microbial World. Chapter 1, Development of Microbiology as a Discipline, and the topic for today's lecture is Louis Pasteur's experiment. But before we go to the actual experiment of Louis Pasteur, let us take a review of the history of microscopy. The first compound microscope was invented by Janssens in between 1519 to 1618. In 1665, Robert Hooke for the first time observed fungi under his microscope. Later on, Leeuwenhoek observed a number of different organisms which he called as the animalcules in a drop of water. Leeuwenhoek communicated all his observation with his drawings to the Royal Society in London. These observations that were communicated by Leeuwenhoek renewed the controversy between abiogenesis and biogenesis. The theory of abiogenesis, that is, spontaneous generation, was stated by Aristotle in 346 BC. He was the first to communicate the theory of spontaneous generation, and his followers proposed the theory so strongly that people started believing in the spontaneous generation. But later on, certain scientists did not believe in the theory of abiogenesis, and therefore they started performing experiments to prove that living organisms are formed from the living organisms. In 1662, Francisco Redi performed the famous experiment of 3 jars and tried to prove that the theory of biogenesis is the theory for the origin of life. But the proponents of the theory of abiogenesis tried to disprove the theory proposed by Francisco Redi by saying that the vital factor pneuma in the air was lost, and that is why organisms did not develop. Right? So the controversy was still there between biogenesis and abiogenesis. Later on, it was seen that when the hay extracts or the meat was boiled and kept aside for a few days, this extracts that were obtained or the broth that was obtained gave rise to the microorganisms. Right? So again, the theory of abiogenesis became a stronger theory. Later, in 1748, an English priest, John Needham, demonstrated an experiment to prove the theory of spontaneous generation. By that time, everybody had known that heat was lethal to the living organisms. So, Needham's experiment consisted of boiling the broth for a small period of time and then cooling the broth in an open container till it came to room temperature. Once the broth was at room temperature, the flask was sealed or the containers were sealed so that no exchange of air took place between the container and the atmosphere or the outside. He observed that after the flask was sealed, a few days later, microorganisms developed in the flask. So, from these experiments, John Needham showed that there was a vital force that produced the spontaneous generation which led to the formation of microorganisms in the broth. In today's days, we know that boiling time was insufficient to kill the endospore forming microorganisms, and that the cooling of the flask that was kept open to the air was the source of microorganisms, and this led to the contamination of the broth with microorganisms, and therefore microorganisms developed in the broth. Needham did not use proper sterilization techniques. Sterilization is a process of removing any form of life. That is, you can say that a particular broth is sterile if it is free from any living organism, whether it may be a spore or a vegetative cell of bacteria, fungi, or a virus. Right? So, Needham did not use proper sterilization techniques, and that is the reason why microbial contamination occurred in the flasks. Needham's experiments were challenged in 1763 by an Italian priest, professor, and a naturalist, Lazzaro Spallanzani. Lazzaro Spallanzani was not convinced by the experiments that were done by Needham. He did not believe that the experimental design was a proper design. So, he modified Needham's experimental design, where Spallanzani put broth in a flask. He sealed the flask, and after sealing the flask, he heated the broth till it boiled. After which, he kept the flask for a few days. When the flask was kept for a few days, the contents of the flask did not come in contact with the air. Neither the contents came in contact with the air when the medium or the broth was being cooled. And therefore, even after a few days of keeping the broth, there were no organisms that appeared in the broth. But the same broth, after boiling, when it was kept open, or after the seal of the containers was broken, within a few days, microorganisms started appearing in the broth. So, Spallanzani concluded that the air that enters in the broth carried the germs or the seeds of those microorganisms, and that the broth cannot produce microorganisms spontaneously. The supporters of the theory of spontaneous generation did not accept the results of Spallanzani's experiments. They argued, saying that heating of the air in the flask destroyed the ability of air to support life. Another point that was raised was the sealing of the flask. Because the flasks were sealed, it prevented the entry of the air which contained the vital factor. And because the vital factor was not available, growth did not occur. But Lazzaro Spallanzani was firm, saying that the air carried the germs or the seeds of the microorganisms, which later on gave rise to the microorganisms in the broth. But the proponents of the spontaneous generation theory wrongly concluded, Spallanzani's experiments and said that air was required for spontaneous generation. So, after Spallanzani's experiments, a number of investigators tried to prove the theory of biogenesis. In 1836, Franz Schulze he passed air through strong acid solutions. So, when air was passed through the strong acid solution, because of the acidic conditions, microorganisms that were present in the air were killed. Right? And this air was allowed to pass in the nutrient solutions that were sterile. So, air was sterile, the nutrient solutions were sterile, and therefore no growth of microbes appeared in the nutrient solutions. Later, in 1837, Theodore Schwann performed similar experiments where he allowed air to enter into the flask with sterile nutrient solution after he had passed the air through a red hot tube. So, when the air passes over the red hot tube, the heat that is generated in the tube is sufficient to kill the microorganisms. And therefore, the air that entered into the nutrient solutions was sterile and thus did not give rise to microorganisms. So, in both the cases, experiment performed by Franz Schulze and Theodore Schwann, the flasks remained sterile even after they were kept for a very long period of time. But the proponents of the theory of abiogenesis did not accept these experiments because they always believed that there is a vital force in the air. And when the air was passed through the acid solution or it was passed through the hot tube, this led to the alteration in the vitality of the air. And because the vitality was destroyed, air did not support the growth of microorganisms. The proponents of the theory of abiogenesis were very strong and they did not accept any of the experiments that were performed till date. In 1850, George Frederick Schroeder and Theodore von Dasch they allowed air to pass through sterile cotton wool, and this air was allowed to enter a flask that contained sterile medium. Now, Schroeder and Dasch did not heat the air. They did not allow the air to pass through the acid solutions, so the vitality of the air was maintained. The cotton wool it helped in sieving the air. So the cotton wool just trapped the microorganisms that were present in the air and allowed the air that was free from microorganisms to enter into the heat sterilized medium. When such air was allowed to enter into the medium, there was no growth that was observed in the flask. Air was not heated and therefore the vitality in the air was maintained. But still, the proponents of the theory of abiogenesis were so strong that they did not believe in these experiments. In 1859, Felix Fauchet claimed that he had designed experiments to support the theory of abiogenesis. He presented a series of papers which stated that abiogenesis was not accidental. and that it was due to the vital force of the material. He also claimed that organic matter, water, air, and the right temperature were required for spontaneous generation. He stated that life originated from the organic matter from the old life and not from the non living matter. That is, the dead and decaying matter, say from organisms, animals, or plants, was the organic matter that gave origin to a new life. So, the controversy between biogenesis and abiogenesis continued for a number of years. In 1859, the French Academy of Science finally sponsored a competition to prove the theory of biogenesis and put an end to the debate on biogenesis and abiogenesis. Louis Pasteur came to know about this competition, and Fauchet's claim that the theory of abiogenesis, or organic matter, water, and the right temperature in presence of the vital force are required for life, provoked Louis Pasteur to enter the competition and finally settle the matter of spontaneous generation. So Louis Pasteur entered into the competition to prove the theory of biogenesis. That is, life originates from other life or from other living organism. To prove the theory of biogenesis, Louis Pasteur performed his famous swan-neck experiment. But before he continued with his experiment, he tried to show that air is full of microorganisms. To prove that air is full of microorganisms, he passed air through the cotton fibers. Right? So these cotton fibers help to filter the air free from microorganisms. He took the cotton fibers and placed the cotton fibers in a sterile medium, which later showed the growth of microorganisms. He also showed that these microorganisms were similar to the bacteria that are found in the putrefying medium. That is, a number of bacteria that are found in protein rich media were identical to the bacteria that were found to be present in the air. It was observed that a number of microorganisms that were present in the air were identical to the bacteria that were found to be present on the dead and decaying animal and plant matter. Thus, he said that bacteria were always present in the air and that they would anytime land on and contaminate any material that is exposed to the air. So, after showing that the air was full of microorganisms, Pasteur entered the contest sponsored by the French Academy of Science to disprove the theory of spontaneous generation. In his conclusive experiments, Pasteur designed a flask with the S-shaped curved tube, which he called as the swan neck. The experiment that was performed by Louis Pasteur can be explained from the figure, in which he took a round bottom flask added the nutrient medium to the flask, and he heated the neck of the flask in such a way that he extended the neck to a shape of S. After having extended the neck of the flask in the S shape, Pasteur heated the broth in order to kill the microorganisms that were present in the broth. After boiling, the broth was allowed to sit and cool to room temperature. There was no cotton plug, there was no heating of the air, and the air was allowed to pass through the S-shaped neck of the flask towards the medium. When air entered the mouth of the neck of the flask, it was full of microorganisms. But the air could pass through the steep curves of the neck, but microorganisms that were laden in the air were settled in the neck of the flask. So, the air that finally entered in the flask was free from microorganisms, and any microorganisms that were present were trapped in the neck before the S shape of the neck of the flask. And therefore, even after keeping the broth for a long period of time, no bacterial growth or no microbial growth was observed in the flask. In the second set of experiments, Louis Pasteur took the same swan-neck flask, heated the flask in order to kill the microorganisms that are present in the broth. After heating the broth, he broke the neck of the flask, the S-shaped neck of the flask, and allowed the flask to set for a longer period of time. So because the S-shaped neck was broken, air with the microorganisms could directly enter the flask. Microorganisms that were present in the air could sit in the medium and grow in the medium. And therefore, after a few days, microbial growth was observed in the flask. In the third set of experiments, he took the same S-shaped flask or swan neck flask, filled it with the medium, and heated the medium in order to kill the microorganisms. He allowed the flask to sit for a few days, after which he tilted the flask in such a way that the medium in the flask came in contact with the microorganisms in the neck of the flask. Again, he erected the flask and kept the flask for a few days, and he found that microorganisms grew in the medium. So, in the first set of experiments, there was no microorganism that was allowed to enter into the broth because all the organisms were trapped in the S-shaped neck of the flask. So, no growth of organisms was observed. In the second set, the neck of the flask was broken, and therefore the medium in the flask was in direct contact with the microorganisms present in the air and thus showed growth of organisms. In the third set, because the medium was allowed to come in contact with the microorganisms that were present in the neck of the flask, the flask did show the growth of microorganisms. Right? So, the experiment ran as Louis Pasteur placed nutrient solutions in the flask, heated the necks in a flame, and made them into curved shape or S shapes. The swan neck flasks that were produced had necks open to the atmosphere. Pasteur then boiled the solutions for a few minutes and allowed them to cool. No growth took place even though the contents of the flask were exposed to the air. Growth did not occur because dust and germs had been trapped in the walls of the curved necks. If the necks were broken, growth commenced immediately. And thus, the controversy between biogenesis and abiogenesis was resolved in 1861 by Louis Pasteur. He also gave us the method by which the media could be kept sterile for a long period of time. If there was spontaneous generation in operation, the broth solution with the so-called unaltered vital capacity of the air would have given birth to the microorganisms. That is, in the first flask, where the air was not treated, it was not filtered, it was not passed through acid solutions, right, would have given rise to the microorganisms. Because the vital capacity or the pneuma of the air was not changed. But although the pneuma was not changed, no growth appeared in the flask. So, with this experiment, Louis Pasteur concluded that the theory of biogenesis is the only theory by which life is originating. So, Louis Pasteur was the one who put an end to the controversy between biogenesis and abiogenesis, proving that the theory of biogenesis is the theory of life. Thank you.